Hello students, so this video is about DNA, this amazing molecule that makes us us and makes all life all life. And so we want to be thinking a little bit about describing the structure of DNA and then relating that structure to its function. As we go through, I want you to keep this question in the back of your mind. What is the role of DNA in coding for the functions of cells? How does it make the protein that makes us? So first let's start with a quick review of nucleic acids in general. So nucleic acids are one of our type of macromolecules. Remember, we have our 4 macromolecules. We have our carbohydrates, we have our lipids, we have our proteins, and then we have the nucleic acids. Nucleic acids are made out of the monomer, or their building block, called nucleotides. Every nucleotide is going to have a phosphate group, a sugar, and a nitrogenous base. If we're looking at DNA, then the sugar is going to be deoxyribose. If we're looking at RNA, the sugar is going to be ribose. Quickly, notice the difference between these two sugars. The only real difference is whether there's an oxygen here, no oxygen here, or 2 oxygens. This one is called deoxyribose because it has 1 fewer oxygen. This one here has 1 more oxygen, so we just call it ribose. For our nitrogenous bases, we have some different options. If we're looking at DNA, it could be adenine, thymine, guanine, or cytosine. If we're looking at RNA, it could be adenine, uracil, guanine, or cytosine. Already, start to notice some of the differences between DNA and RNA. Now let's see how the polymer goes together to form these nucleic acids. There's going to be some stronger bonds involved and some weaker bonds involved. Let's start with the stronger bonds. The phosphate group of one nucleotide is going to form a covalent bond with the sugar of the next nucleotide. This bond here is going to be fairly strong. It's called a phosphodiester bond. This is going to together form what we call the sugar-phosphate backbone. This term backbone should convey that this is fairly strong, fairly stable chemically. Then there's going to be a second set of bonds. The bases are facing here in the center of this molecule. And every base is going to have a base pair that it can form based on the chemical structure. Adenine is always going to pair with thymine, and guanine is always going to pair with cytosine. Notice the adenine and the thymine are going to form 2 weak hydrogen bonds. The guanine and cytosine are going to form 3 weak hydrogen bonds. Based on the order of these nucleotides, whether it's A, then C, then C, then G, or whatever the order is, that is how these nucleic acids get their function, which is the storage and transfer of genetic information. When we're looking at our complementary base pairing, so T going with A and C going with G, there's going to be a couple different structures that we notice. First, we have purines. Purine is the name for a nitrogenous base that has 2 rings with it. Notice we have adenine here with 1, 2 rings, and guanine with 1, 2 rings. Those are both our purines. Then we also have our pyrimidines. They're nitrogenous bases with 1 ring, so that's thymine and cytosine. Notice there's always going to be a pair between 1 purine and 1 pyrimidine. That's going to keep the length of these things, or the width of this DNA, constant all the way down. Another thing you'll probably want to notice when you notice how this macromolecule is coming together is there's going to be one side that looks a little different than another. We call one the 3' side and one the 5' side. That 5' and 3' just refers to the number of the carbon in the sugar and which direction it is forming. Notice when we come and we form these base pairs, the 3' on one base pair is going to be paired on the 5' of the other. That's going to form the structure that we call anti-parallel. They're running next to each other, but in opposite directions. Notice as we're forming this covalent bond, we're going to have some phosphorylation in order to do that. And then these hydrogens are what's happening in the middle. They all come together based on the chemical structures, their base pairs. Okay, once we have all those things in this unwound structure together, then there's going to be some additional interactions between some of the side groups. That's going to cause this whole thing to twist into that double helix shape that we think of. If we took this double helix shape and we just stretched it out in a single cell, then it's estimated that just a single bit of DNA could reach 2 meters. It's a really, really thin, tiny molecule. But in order to fit it into a tiny, tiny cell, we've you have to package it really, really tightly to fit it inside the nucleus. So we can have the DNA here. It can be bundled up with histones and then coiled again and then coiled again and then coiled again, and eventually we get to chromosomes. Let's look at those structures. Here we have our DNA. Notice the width of this thing is only about 2 nanometers. It's tiny, tiny, tiny. Then we have this thing called histones. Histones are proteins, and if you are a eukaryotic cell, then the DNA is going to wind around these proteins kind of like beads on a string. Also think of it like thread wrapped around something that allows it to get really, really close. Then those histones, together with the DNA, which is called a nucleosome, are going to wrap around each other a couple of different times to form these really strong, super coiled loops. Eventually those loops are going to be coiled upon each other enough so they get towards the centrosome. This is eventually going to form that chromosome, which is going to allow separation during mitosis and meiosis. Okay, so already we started talking a little bit about the differences between two different nucleotides, DNA and RNA. DNA is going to be the one that's responsible for longer-term storage of information, and it's a little different than RNA. RNA is going to be responsible for creating more actively the proteins that make us us. So if we were to compare the structure of RNA and DNA, there's going to be some similarities and some differences. For example, both have a 5-carbon sugar. However, in DNA, the 5-carbon sugar is deoxyribose. In RNA, it's ribose. In DNA, we're going to have 4 nitrogenous bases. Same with RNA. However, in DNA, there's cytosine, guanine, adenine, and thymine. And then in RNA, there's cytosine, guanine, adenine, and uracil. DNA is going to form that double helix idea of that double-stranded molecule. RNA is found as single-stranded molecules. So single-stranded doesn't always mean straight. Sometimes it can fold a little bit on each other, but it's generally single-stranded. Let's look at a couple examples of this. Again, we see, okay, here's the DNA side with our deoxyribose, our T, and our double-stranded. Where over here on the RNA side, we have our ribose, our U, and single-stranded. All right, guys, so there's a little background on some of the structure of these things. Later, we're going to go a little deeper into how that structure allows proteins to be built, and then how those proteins form us. Hope that helps.